Hello, and welcome to the latest METTLER TOLEDO webinar. My name is Ian Haley, and I am a Senior Technology and Application Consultant for our Particle Systems Characterization group. Today's webinar is entitled Understanding Disintegration and Dissolution for Improved Solid Dose Formulations Development. Here is an outline of today's webinar. I will talk about how process measurement tools can be used to aid solid dose product development via dissolution studies. This is intimately related to the principles of quality by design, so I'll start by introducing quality by design in dissolution and disintegration processes. I'll carry on by running through 4 different case studies, which highlight how analytical tools have been used to gain understanding in dissolution and disintegration studies. I'll start with a very quick introduction to dissolution. Dissolution tests on tablets are a regulatory requirement, and these tests are typically carried out in a standardized apparatus to a set procedure, and the aim is to ensure that the correct amount of drug active is released into solution from a tablet. So, what is wrong with dissolution testing? Well, whilst the test itself is invaluable, the results tell you nothing about why the tablet has passed or failed. Let's take a look at the FDA stance on dissolution testing. This can be understood by referring to a paper by Mohib Nasser to the Advisory Committee of Pharmaceutical Sciences in October 2005. They pointed out that the established procedure for dissolution testing did not really allow for adequate root cause analysis when products failed the test. In their summary, they pointed out that in a quality by design approach, it should be possible to gain better process understanding that links manufacturing controls to specifications and critical quality attributes. And this in turn will lead to the desired performance of the drug product. In addition, in some cases they pointed out that critical quality variables, such as particle size, may be more relevant to achieving good quality than a simple dissolution test. Now, an example of an incomplete approach to understanding why tablets fail a dissolution test is given here, and it concerned Andrix Pharmaceuticals of Florida. Importantly, the FDA pointed out that their investigation into why tablets had failed the dissolution test did not address the real reason why certain capsules didn't dissolve adequately or why they took longer to disintegrate. Importantly, they pointed out that the inconsistency was possibly process-related, and that would have required the investigation to be extended outside of the lab to determine if the root cause was indeed process-related. So, why do the formulation and the process impact on dissolution? Let's suppose that we're trying to optimize tablet dissolution. What factors can cause variability in the tablet dissolution process? Well, the most immediate thing is any variability in the tablet press itself. But that in turn could have been caused by upstream processes. For example, there may have been inconsistencies in the blender. There may even have been inconsistencies in a mill taking unmilled material before blending. In turn, there could also have been inconsistencies in any fluid bed conditioning of granules before they were finally milled and blended. Going further upstream, there may even have been batch-to-batch inconsistencies within the granulation process itself. And that, in turn, could have been caused by problems with either the API, the excipients, or even the binder. So let's have a look at the kind of reasons that may give inconsistencies in each of those unit operations. In the tablet press, we may have poor flow properties. That in turn could lead to some size-dependent segregation, and that will lead to variable tablet properties. In the mill, we may be milling to an incorrect size. We may even have incomplete milling, leading to a much wider than expected size distribution on the outlet of the mill. In a fluid bed, our drying conditions may be too aggressive, and that could lead to an excessive attrition, i.e. producing too many fines. We could also have segregation within the mass in the fluid bed. Within the granulator, we could have incorrect endpoint distribution. In other words, too many fine particles, too many coarse granules, or even some kind of bimodal distribution. We could get agglomeration of the API crystals within the granulating batches. And we could have problems with wet massing or even moisture addition, leading to inconsistent granule density. Finally, we may have inconsistent raw materials. That could lead to segregation in the feed. We could have a variability in API particle size, or even impurities contained within the API. All of these regions can lead to variability within the process and thus enable it to have difficulty optimizing the tablet dissolution process. So, if we look at this in terms of the key elements of a quality by design approach, in QBD, we first need to gain process understanding. In other words, we need to identify how a given unit operation links to downstream process or product parameters. By carrying out a number of design of experiments, we can characterize those process limits when we develop and scale up our process. And thus we can map the process to identify those critical product and process parameters. Once we've identified those critical quality parameters, that enables us to take a risk management approach where we can thus isolate the root cause of any process upset. And by using process analytical tools, these are enabling technologies that allow formulators and engineers, gives them a rich source of information to make better decisions faster and thus to build quality into a process in an effective way. So, let's have a look at dissolution and process analytical technology. Dissolution testing with in-situ particle characterization is a very powerful tool in a quality by design regime. The METTLER FBRM probe can link tablet disintegration and dissolution mechanisms to upstream formulation process upsets and to the initial API and excipient particle size. If we combine FBRM with other techniques such as UV-VIS or infrared spectroscopy, those can be used to track the solute concentration in real time. In other words, they can be used to track the level of API that has been released into solution. Combined, these PAT tools can significantly speed process development by understanding if dissolution inconsistency is due to the formulation, the manufacturing process, or the raw materials. And here's a picture of an FBRM probe installed in a standard Distech dissolution apparatus. I'd just like to quickly explain the METTLER FBRM instrument. FBRM stands for Focused Beam Reflectance Measurement, and this technology is a probe-based instrument that's installed directly in the process. In this case, it's installed directly in the dissolution apparatus, as we can see in this photograph. FBRM tracks any real-time change to particle and droplet systems as they naturally exist in normal process conditions. FBRM works by tracking changes to the chord length. This is a function of particle dimension, particle shape, and also the population of particles. By measuring directly in the process, we eliminate any need for offline sampling and analysis. This enables faster methods of designing and optimizing particulate processing because we can perform many experiments in a short space of time without waiting for offline results. It also provides in-situ documentation of critical process parameters, and this in turn facilitates, for example, quality by design information to support FDA filings, root cause analysis to determine why specific batches fail QA or QC specifications, and finally, it allows much more efficient scale-up and tech transfer by virtue of using batch-to-batch fingerprinting and reliable endpoint detection. So, let's move on to the first case study. And in this case study, we can see how FBRM has been used to help understand the disintegration kinetics of tablets, and that in turn has helped lead to an appropriate risk assessment. So, FBRM is inserted into the standard dissolution apparatus, and because it measures particles, it will track changes to any non-dissolved excipients. Thus, as the tablet disintegrates and breaks up, FBRM will track the change to the number and size of particles in the liquid. In addition, a fiber optic technique, such as UV-VIS,
has also been used to track the level of API in solution. And these two in combination can give us a wealth of information about the mechanism and kinetics of disintegration and dissolution. In this first case study, this is work carried out by Jonas Johansson of AstraZeneca in Sweden. First, a little bit of background information. The API release is primarily governed by erosion of the gel-forming agent in the tablet, and thus is controlled by the disintegration kinetics of the tablet. In other words, the tablet needs to break up, allowing the API to be exposed to the liquid, to the aqueous solution. Now, it's also possible that diffusion of API directly from the tablet can also play a part, and that in turn can speed up the dissolution process. So, where the FBRM signal and the UV-VIS signal follow similar trends, we can say that the rate of release of API into solution is primarily controlled by the disintegration of the tablet. In other words, the rate of diffusion is modest. Where the FBRM data and the UV-VIS data follow rather different trends, we can thus say that diffusion may be significant. Now, I also need to explain a little bit about the biopharmaceutical classification system, or BCS. This typically classifies drugs into 4 different classes, and it's based on their relative solubility versus permeability. So, just to reiterate the different mechanisms for tablet breakup. This is illustrated by this diagram here. So when a tablet breaks up, it initially breaks into large fragments, and these in turn can further disintegrate into individual particles. And there are 2 mechanisms by which the API can be released into solution. One is a direct result of the disintegration process. The smaller the particles and the greater their number, the more API particles are exposed to liquid and therefore the faster they dissolve. But there's also the diffusion process, whereby water can enter the tablet or the large fragments and therefore solubilize the API directly. So let's have a look at an example where a BCS class 1 drug, that's one where both solubility and permeability are high, has been dissolved. Now, for a class 1 drug, we expect that the dissolution and disintegration kinetics should be approximately the same. In other words, the rate of dissolution of API is directly controlled by the breakup of the tablet into fragments. In the graph below, 3 different statistics are plotted. The red line shows the proportion of API that has been released into solution as measured by the fiber optic technique. The blue line and black line are 2 different FBRM statistics. The black line is tracking the number of small particles as they come into solution, as they appear, and the blue line is tracking the number of large particles. And as we can see, the rate of dissolution very, very closely follows the rate of appearance of large particles, showing us that the solubilization of API is directly controlled by the disintegration of the tablet into large granules. Now, it's important to remember that changing the design space can impact the dissolution properties. So, for example, in the previous case, dissolution was carried out at low pH. And here, in the graph on the left, we can see that the UV-VIS data, the red line, closely follows the FBRM data, the blue line, showing us that disintegration is a rate-limiting step. However, the graph on the right shows a similar study carried out at much higher pH. And here we see that the dissolution profile is now different to the particle profile. And that tells us that at higher pH, the API solubilization has now become the rate-limiting step. So, 2 simple experiments and show us that real-time particle understanding could significantly speed up the development time by very quickly enabling a user to uncover what the rate-limiting step is. And in this case, it's pH. Now let's have a look at some data from a BCS Class 2 drug. In this case, examples of good and bad tablets were prepared and monitored by FBRM and the UV-VIS fiber optic technique in the standard dissolution apparatus. The results are shown here. The FBRM data is represented by the black line for the good batch and the red line for the bad batch. And in both cases, we're tracking the average size of the particle fragments. As we can see in the bad batch, we have a higher average size of fragments throughout the dissolution and disintegration of the tablet. If we then look at the UV-VIS data, the bad batch is characterized by the green profile and the good batch characterized by the blue profile. And as we can see, the bad profile results in a lower amount of API released into solution at a somewhat slower rate. In other words, we can conclude that a bad tablet is characterized by a tablet that is brittle, and it quickly breaks into rather large particles, and these are not well disintegrated, and these do not release API into solution as effectively as a good batch where we have smaller particles and a quicker rate of release. In other words, and this is just the same data looked at the earlier stage in the process, FBRM is able to immediately identify the direction investigators should take to uncover the root cause of a dissolution inconsistency. And clearly here, the dissolution inconsistency has been caused by the size of fragments when a tablet breaks up. In this case, the root cause was identified as due to the type of filler used in the process. A new filler was selected, and thus the problem was rectified. And as we can see, we now have data from two different types of batch of tablets, where in both cases the average size of fragments as tracked by FBRM is the same, and also the rate of dissolution of API into solution as tracked by the fiber optic technique is also the same. And this is a very simple example of how a quick study using FBRM and other techniques can save months of development time and ensure that the formulation design is understood and of course can avoid processing questions from the regulatory body. In the next two case studies, I'll show how an investigation of tablet properties led to a better understanding of the allowable design space. In case study 2, this is taken from work by Vertex and was presented at the AAPS meeting in April of this year. In this study, tablets with different degrees of hardness were investigated in the standard dissolution apparatus. On the left is a graph showing the dissolution profiles for 5 different tablets of 5 different hardnesses. As we can see, the tablet with the lowest hardness gives a greater amount of API released into solution. The tablet with the greatest hardness results in a lower amount of API into solution. At the same time as the dissolution was monitored, FBRM was also used to monitor a change to the average size of fragments as the particles broke up. And this data is shown on the right-hand side for the corresponding 5 tablets of different hardnesses. As we can see from the FBRM data, the tablet with the lowest hardness, that's the red line, yields an average particle size that is significantly lower than the tablets of greater hardness. And in fact, the tablet with the greatest hardness, that's the black line on the right-hand graph, yields the highest average size of fragments. In other words, in this study it was shown that the tablets with the higher hardness disintegrate into larger granules, and that in turn results in a slower release profile of API into solution. In case study 3, we show a similar study carried out by GlaxoSmithKline. In their study, they looked at 2 different types of tablet, a tablet made with low compression and a tablet made with high compression. The API release data from the dissolution test is shown in the left-hand graph. Here we can see that it was the high compression tablet which resulted in a greater release of API into solution. The low compression tablet resulted in a lower rate of API release. This is the opposite of the findings from Vertex. However, we can gain an understanding of why this occurred if we look at the corresponding FBRM data, and that's shown in the graph on the right. Here, 2 different chord length distributions are plotted. The red distribution shows the data from the high compression tablet. The black distribution shows the data from the low compression tablet. And in this case, we can see that the low compression tablet yielded an average particle size which was greater than the high compression tablet. In other words, the higher compression resulted in a smaller average size, and that helps explain why in the GSK case that resulted in a greater rate of release of API into solution. So to summarize, in the Vertex example, a higher compression force results in larger granules, and that in turn leads to a slower dissolution rate. However, in the GSK example, a higher compression force results in smaller granules, and that thus leads to a faster dissolution rate. Thus, monitoring granule dimension during dissolution can help understand inconsistencies and will enable the formulators to target a consistent dissolution profile with each new formulation step that they add. I'd now like to move on to the final case study, and here I'll show how dissolution tests have been used as a mechanism to uncover root cause analysis. This is from work carried out by Amgen and was presented at the AAPS meeting in 2007. In the Amgen work, a comparison was made of granule disintegration and tablet disintegration for a formulation that had been granulated with two different water levels, a high water level, HWL, and a low water level, LWL. So for this test, two different granulation batches were prepared in a high shear mixture. They contain the same API and insoluble excipients, but these two different levels of water. These two different batches were then made into tablets using a rotary press. Then, samples of the granules and the corresponding tablets at these two different water levels were dispersed into a USP dissolution apparatus. The size distributions and the average size was monitored over time during the disintegration using the FBRM instrument. So, in this test, this shows the data from the dissolution of the tablet. And as we can see, the high water level tablet results in a lower average size during the disintegration and dissolution process, whereas the low water level tablet results in a higher average size of fragments as the tablet breaks up. If we have a look at what happens at the end, these are now the corresponding chord length distributions from the end of the dissolution process. And we can see that the high water level tablet has indeed yielded a smaller size distribution than the low water level tablet. Thus, the water content during granulation clearly has an effect on the way in which the tablet breaks up. We can now look at the corresponding data just from the final blend of granules. So, this data from FBRM is seen in the graph here. So, when the batch of granules with the high water level was dissolved, that resulted in a larger average size than when the batch of granules from the low water level was actually dispersed into the dissolution apparatus. In other words, the high water level granules remain much larger than the low water level granules. So let's have a look at that and combine the data from the tablet and the granules. So, if we look at just the low water level data, we can see that the data from the tablet and the data from the final blend of granules is not identical. And this suggests that there is incomplete disintegration of the tablet. If we look at the equivalent distributions at the end of the experiment, there is a difference. The final blend of granules yields a slightly smaller size than the final size distribution of the tablet fragments. The hypothesis is that the original low water level granules undergo extensive fragmentation and they so form larger granules during the compaction into the tablet. Let's now have a look at the data from the high water level experiments. Here we can see that as we approach the end of the experiment, the high water level granules, the final blend, and the high water level tablet reach the same average size as measured by FBRM. If we look at the graph below, we can see that both the final blend granules and the tablet at the end of dissolution yield a very, very similar size distribution. In other words, the suggestion is there has been negligible granule fragmentation during the compaction process. So in other words, a high water content leads to very little change in the granule size from the original blend into the tablet. So, I hope I've shown in these 4 case studies that if we investigate the fundamental mechanisms that cause tablets to fail the dissolution test, we can gain some very valuable insight. So just to summarize, FBRM has been used to understand the impact a formulation step has on tablet disintegration and on dissolution mechanisms. We've used the technique to identify the root cause of dissolution inconsistency, and that has led to a better understanding of the process. In turn, FBRM can speed process development by providing an immediate understanding that allows formulators to make better decisions faster. And this can save months of development time. It will allow us to target consistent final products and, of course, ensure that formulators have the correct answers for any regulatory questions that might arise. Finally, I'd like to acknowledge the following people who have helped significantly and contributed to the data that I've presented in this webinar. That concludes the webinar, and now there'll be a short questions and answer session based on the questions that you have put to me whilst I've been talking. Thank you very much for your attention.